Hello, and welcome to this MEGIN Masterclass. Thanks for joining us. My name is Nicole Suri, and today we're delighted to be joined by Hanna Renvall, Assistant Professor in Translational Neuroimaging at Aalto University and Helsinki University Hospital. There will be a Q&A at the end of this presentation, so please feel free to submit your questions as we go along using the button on your screen. If you'd like to revisit any part of the session, the webinar will also be available on demand on the MEGIN website tomorrow. Now, without further ado, it's my pleasure to hand things over to Hanna Renvall, who will officially begin today's masterclass. Thank you.
Thank you. Nice to be here today. So my title today involves predicting dementia. So let's go directly and do the talk. So dementia is a huge burden to the individuals themselves and to their close ones, and also to the society. Mild cognitive impairment, or MCI, refers to a stage between normal brain aging and clinical dementia. And it is a condition affecting currently more than 10 million people in Europe alone. With the aging population, this number is predicted to double within the next 20 years. Subjects with MCI form a particularly interesting group. We know that they are in a significantly increased risk of developing dementia, especially Alzheimer's disease, within the next 5 years. But many of them also stay stable or even get better, as you can see from the curves on the right side of the figure. For trying to slow down the cognitive decline and for providing the needed support for these subjects, we should find the ones in the biggest risk of progression as early as possible. The diagnosis currently mainly is based on clinical symptoms, so biomarkers for early diagnosis are urgently needed. To this end, MEG and EEG can be highly informative. In Alzheimer's disease, synaptic level dysfunction appears to be one of the earliest changes at the neuronal level, and it may precede the clinical symptoms by several years. The synaptic changes could potentially be pinpointed by MEG and EEG. This picture, which is from a review written by Fernando Maestro and his co-workers, describes the hypothesis behind this idea. At the neural level, Alzheimer's disease is characterized by accumulation of amyloid bodies outside the neurons. The amyloid bodies would have a toxic effect at both pre- and prosynaptic level and cause initially neuronal hyperexcitation and later silence the neural activity. These changes, in turn, cause excitation-inhibition imbalance at the local neuronal circuits, and finally, they will lead to changes at the brain network level, which would then cause the cognitive symptoms that we see in these patients. Indeed, there already exists data suggesting that MEG and EEG can truly provide early markers of mild cognitive impairment. And Nakamura and co-workers demonstrated already some years ago that the regional spectral patterns of resting state activity differ between controls and MCI patients. Subsequently, Sandra Pusil from Fernando Maestro's group in Madrid showed in their longitudinal study that MEG functional connectivity combined with machine learning can predict pretty well which of the subjects they had been following suffer from progressive disease and which do not. So on the right side of the figure, you can see those subjects at 3 years which were predicted to be progressive and which are stable on the basis of the early MEG results. And as you can see, the machine learning algorithm was pretty good in differentiating these two groups from each other. Well, our Italian colleagues used EEG, functional connectivity measures of EEG, and combined it with genetic susceptibility, and namely ApoE4 status, which is known to reflect, be one of the biggest reflectors of Alzheimer's disease risk, and they acquired excellent classification accuracy of 0.97. So it kind of appears that there's a lot of butter on the bread, and maybe even that everything is already done. But really, when these measures have then later on been taken to other patient cohorts, the results have not been that well generalizable. So it became evident that at this point we need large, unified data sets for extracting the data features which would be generalizable and most informative of the risk of cognitive decline in these subjects. So what we did, we put up an AI Mind study. We got funded via EU Horizon 2020 program. And the idea here has been to collect altogether 1,000 participants suffering from mild cognitive impairment and follow them with 128 channel EEG measurements, and here in Helsinki also with MEG measurements over 2 years at 8-month intervals. The goal is to use machine learning approaches in finding the best EEG and MEG features that are informative of network disturbance in these subjects. And at the next stage, combine this information with genetic and blood biomarkers, and also with very versatile cognitive tests that we have collected from these patients in order to form the best overall estimate of the dementia risk in these subjects. We are now running the last visits of these participants, so we ended up with 1,080 subjects altogether, 230 of them in Finland, and this 2 years measurement follow-up time starts to be in the end. So, well, here in Helsinki, we have been doing EEG and MEG measurements at the same time, while the other clinical sites have only focused on the EEG activity. So why to measure both? We believe that using both can benefit us in source localization. And anyway, MEG can be very helpful in interpreting the EEG findings, as well as help in fine-tuning the EEG measurements and analysis later on. In front of this audience, I would also like to emphasize that it's a big difference in practice for the participant and also personnel to do the MEG measurements, which take, let's say, 15 minutes of preparations, while with 128 channel EEG, the preparations can easily take 45 minutes. So it requires a lot of manual work from the personnel, and usually the subjects start to be quite tired at that point, which is not really optimal. But what do we then want to achieve? So we want to achieve faster diagnostics in order to get into more effective interventions. So this slide kind of describes the long-term goal. So when the first cognitive symptoms would appear, patients would, in addition to the clinical examination, go through an MEG or EEG measurement, and the data would be combined with information from cognitive tests that could be then conducted even with the mobile device, and maybe blood samples, blood biomarkers could be added to the analysis for forming an early risk assessment. This combined information could then be used for risk factor recommendation. We know that with controlling of blood pressure, blood sugar level, cholesterol levels, we can significantly slow down also the progression of Alzheimer's disease. And importantly, we may be able to provide well-screened candidates for future medications in the field. As some of you may know, after 20 years, we are now at the stage that we are introducing new drugs at the clinic to Alzheimer's disease to slow down the cognitive decline. And these medications are very expensive, and they can also bring highly problematic and significant side effects to the subjects. So we need tools for the best possible patient selection. So even though we are still in the phase of collecting data in the project and the final analysis and results are yet to come, we have already learned several things from the study. So in the following, I will show you that MEG and EEG, first of all, provide stable features also in MCI subjects that can be clinically used. I will show you that MCI subjects' rhythmic activity does differ from controls, and the biological risk of Alzheimer's disease is also reflected in MEG responses. So even before we can answer our main scientific questions in the project, the collected data has already shown to be highly fruitful and useful for many questions. For example, it's not known before how stable are these measures we want to look at this particular patient population. And for example, how much data is needed in the end for providing good data features for the analysis. We very seldom have this size data sets to work on, and these kind of questions are in the end clinically very valid. These are not healthy 25-year-old university students. These are 60 to 80-year-old people, some with an emerging neurological disease. So we have, for example, already addressed questions of stability within measurement and between measurements. For example, for typically used power spectrum density measures such as alpha and beta activity measures, and evaluated their stability within and between sessions. And as you can see in this figure, we have an excellent alpha peak frequency, beta peak frequency stability within measurement sessions, so between 5 minutes runs within the same day, showing that these features are indeed well reproducible. The same applies for the relative power at these particular frequencies in these subjects, shown here also using this intraclass correlation coefficient, which is almost 1 for both alpha and beta relative power, showing an excellent stability. We are now working on asking how much data is needed to produce these stable features, because of course in clinic it makes a big difference whether you can have good measures in a couple of minutes compared to tens of minutes of measurements. We have also been comparing data across 8 months apart measurements. We don't expect the features to decline a lot during this short period of time. So as you can see, again, we can have a very good retest stability in these very commonly used features like alpha peak frequency, alpha relative power. So in conclusion, we do see excellent within and between session stability of commonly used power spectral features also in mild cognitive impairment subject, demonstrating that these are clinically feasible measures and could be used also in bigger patient cohorts. In Finland, we also have a subgroup of subjects that fulfills rather subjective cognitive impairment criteria than mild cognitive impairment criteria, meaning that they don't really show signs of cognitive impairment in objective testing. Here we have used them as a kind of a control group and try to check whether we can, for example, repeat the earlier findings on certain frequency bands. So basically we have been using machine learning based on support vector machines to assess the relevance of different frequency ranges and also regions of interest of MEG channels for classifying the MCI and SCI subjects. And for this, we used 10 minutes of resting state MEG data and looked at the power spectral densities over specific channel selections. And indeed, the earlier findings are quite well reproduced here. So what you here see, that is a frequency-based decoding over occipital and central channels in these subjects. And these frequency ranges, which are pinpointed here with the blue arrow, show those frequencies where these subject groups differ clearly from each other. So the previously described fairly low frequency ranges below 10 Hz, also around 10 Hz, and interestingly also around 20 Hz can be produced with a fairly simple measurement and analysis, showing that these are indeed informative frequency ranges already in a fairly simple power spectral density analysis, again speaking for their clinical utility. I have to admit that when one has such a big and versatile dataset in hands, it kind of feels like being a kid in a candy store. You don't even know where to start from looking at all the data things. To me, as a former clinician, one evident question is that what part, if any, of the MEG activity reflects the different biological risk factors of Alzheimer's disease that we can get, for example, from the cerebrospinal fluid markers that are nowadays considered the clinical golden standard of Alzheimer's disease risk in many countries like Finland. We had initially the cerebrospinal fluid information available from 69 of our subjects. We also have information of the best characterized genetic risk of AD in these subjects, namely the ApoE gene status. We looked at how well does the CCF and genetic status for the ApoE correlate within this fairly small subject group, and indeed we do see a clear correlation between the cerebrospinal fluid risk and the genetic risk. So the ApoE risk and going down of certain markers in the cerebrospinal fluid correlate significantly, showing that we have a very good population in our hands. What we then did, we looked at some of the MEG markers in this fairly small group of subjects, but already here we can see that this cerebrospinal fluid risk is reflected in the MEG beta power, so around 20 Hz. Speaking for that, we indeed have powerful markers in our hand. I think this is highly promising, but of course now the next question will be to look at what will be the additive value of MEG to these other known risk factors. I will now switch a little to not completed, but a bit different direction, as all the measures that I'll be now describing to you are like from passive resting state MEG, EEG measurements. But here in Helsinki, we have been all the time thinking that also it could be really interesting to measure some tasks, or measure these subjects under certain tasks, while we see all the trouble in getting them under the EEG and MEG devices. So we also have come up with the task with the idea of whether we could kind of boost some of the differences in the brains of these subjects with well-defined tasks. First, we have been looking at semantic processing in our subjects. There is previous evidence from MEG mainly that abnormalities in the so-called N400 or later components in EEG in lexical semantic tasks may be indicative of a memory disorder. So to this end, part of our participants performed a task where they were asked to touch whether two written words presented to them were related, like banana and orange, or unrelated, like banana and police. After each target word or target verb pair, the participant needed to make a decision and respond with a button press. So this is a kind of a modification of the classical N400 paradigm. So what do we expect here? So we expect that the responses to unrelated or incongruent targets would show stronger activity in the brain than the related congruent targets. And we assumed this congruency effect, where the responses to related and unrelated target words differ, would be bigger in our participants with the developing memory disorder. So like shown on the right side of the figure. The PI of this part of the research project has been Doctor of Technology Mia Liljeström from Aalto University and Helsinki University of Hospital. So we set up this project in 2 different stages. The first aim was to understand how lexical semantic processing differs between healthy young adults and healthy elderly adults in order to make sure that this is a reliable marker of lexical semantic processing that would work also in elderly population in general. And then the second aim was to apply this marker in our MCI participants. But let's first look at our healthy participants. So we measured altogether 50 participants. Half of them were young, around 25 years old, and the other half around 65 years of age. We excluded any significant cognitive impairment from these subjects with cognitive testing, like Montreal Cognitive Assessment, and the stability of these measurements was assured by conducting the MEG measurements twice, 1 to 2 weeks apart. So what did we find? We were able to reproduce the typical N400 effect. So incongruent target words yielded strong N400 responses in the temporal cortex of young adults, as you can see in this picture, around 300 to 400 milliseconds after the presentation of the word pair. Well, how about the healthy elderly participants? We didn't see exactly the similar kind of effect. So they showed weaker activation in the temporal cortex around 300 to 600 milliseconds, but they had fairly strong frontal cortical activity, suggesting that they might be having different kind of way of doing the task, maybe relying more on the frontal cortex functions. But they anyway showed retained performance, so they performed very well in the task, similarly to the young subjects. So we have now established that the task we have chosen does give us reproducible results, and we also noted this clear shift towards activation of the frontal cortex in older adults, demonstrating that we do need to have age-matched controls in our clinical studies. And we cannot just directly translate results from young healthy participants to an elderly population in general. Well, next we wanted to develop a marker of semantic processing that we could apply to MCI subjects. So first of all, even though we have now demonstrated that the group effects are reproducible and also elderly subjects in this task in healthy population, but we also noticed that the MEG responses are highly individual. So different people have different kind of brain activity, as you all there know as well. So we wanted to extract the individual effects from the subjects. So what did we do? We constructed an individual machine learning model that aimed to decode at an individual level whether across trials a participant was seeing a related or unrelated word. So basically, if the lexical semantic processing was intact in an individual, there should be information in the MEG signal such that we can code it with high accuracy. So in other words, as you can see on the left side of the figure, if there would be a strong congruency effect in the MEG signals, there should be reflected as a high decodability from the MEG responses, while weak congruency effect, so not much difference between the different word pairs, would be reflected as low decodability. So this was our hypothesis, and we used logistic regression on sensor level data. And here are the results. So these traces show the group averages of the time resolved decodability over the young and old participants. So the congruency effect was indeed decodable in both young and old participants. And you see again that the importance of the temporal cortex was emphasized in the young participants, and in the older subjects, this decodability more reflected frontal activation. What was also interesting was that this decodability of the congruency effect was related to task performance. It correlated with the reaction times in these tasks in these participants. That is, participants who had good decodability, meaning that they had more information present in the MEG signal about the semantic congruency, they were faster, whereas participants who had low decodability or weak congruency effect, in other words, were slower in performing the task. And this was true for both young and old participants. So we now have a task that gives reproducible results, and we decided we have now kind of decided we have a good marker of semantic processing, correlates with performance. So how about this same task in our MCI cohort? So we recruited 70 of our AI Mind study participants to this part of the study. So of those, 23 fulfilled the subjective memory concern requirement, and 44 fulfilled mild cognitive impairment criteria based on a bit looser and then also a bit stricter criteria, which now is defined here as MCI minus and MCI plus groups. And then let's look at the similar kind of curves about the decodability. And indeed, the decodability of this congruency effect in this semantic lexical task does differentiate our MCI subjects from the SCI subjects around 600 to 700 milliseconds, shown here with this shaded area in the curves. And also in these subjects, this result correlates with task performance. So it appears that semantic processing is weaker or more variable across the trials in our MCI than SCI subjects. Now, of course, important question is that does this finding predict the cognitive changes in these subject groups? And this is now something that we are currently working on. So these subjects have now visited us for the second time. We have new MEG measurements, we have new cognitive data, but we haven't had time to look at the results yet. But maybe next time I'll be on this channel, I can tell you about that. Well, I want to conclude this talk to the obvious question. So how far are we from the routine clinical use of MEG or EEG in dementia diagnostics? Well, maybe not that far. In Amsterdam, MEG is already in clinical use in aiding differential diagnostics in memory disorders. This figure is from Alisa Gauss and Go workers' article, where they describe their clinical pipeline, which already includes resting state MEG and machine learning approach to give a prediction that the clinician can then use for forming their opinion. But at the moment, this is more informative for kind of bit more advanced stages of dementia, but already in use in differentiating between different dementia types. But overall, the question about the earliest markers are still in the air. And so we are still trying to decide what are the best electrophysiological features. But I hope that I have now convinced you that we have many good candidates already in our hands to work on. So to conclude, MEG combined with machine learning or AI can provide early non-invasive biomarkers of cognitive decline. Biological risk factor of Alzheimer's disease is reflected also in MEG features, but large data sets like this are needed for establishing the clinically most useful and most stable data features. And the clock is there just to remind us that timing is important also when we are diagnosing neurodegeneration. So we should be as fast as possible, also with these patients, to be able to provide the best possible care and follow-up. So thank you for listening, and then I would like to thank our fairly big group here in Helsinki University Hospital, who have been responsible for measuring these subjects, for doing the neuropsychological evaluations, different kind of interviews, and so on. And of course, I also want to thank the whole AI Mind Consortium, and of course also the many sites that have given us monetary support to conduct these studies. Thank you.
Thank you. That was a great masterclass. So now we'll move to our Q&A session. Anyone online, we'd encourage you to write your questions in the chat as well, please. But we've got our first question, which is, you appear to do many of the analysis at the sensor level. Do you think that it is enough, or do you think going to source level would provide some needed extra information?
Yes, that is true that we are, the data that I showed today was mainly at the sensor level. And this is also something that we have been thinking for some while, that because as we try to find clinically informative measures, then also we would want to have them as simple form as possible, if that's enough. And in that sense, simple sensor level measures would be really much very appealing. And indeed, many of the things that we have been working on appear to show really nice effects already at the sensor level. But we are currently running these similar kind of things also at the source level, and in figuring out, do we get extra information at the source level, which would be critical for the diagnostic accuracy and so on.
So we have another question in the chat here. Were your ICC values from MEG or EEG? Will you test whether MEG data provides additional information about dementia beyond EEG? In brackets, there has been a lot of work for many years using EEG plus ML AI for dementia, but not sure it has produced much clinical value yet.
Yes, thank you. The values that I showed you today, they were from MEG. We have the similar values from EEG, so we have done the same comparisons there as well. The MEG ICC values are a little bit better, at least at this stage, maybe reflecting also that the MEG measures are a little bit less noisier and so on. But basically the EEG ICC values are also very good. We are actually a bit kind of struggling that how to then bring this forward, because of course comparing MEG and EEG is not that simple, and there can be many confounders and so on. But indeed, this is kind of our goal, so that to test that, do we get additional information from MEG with respect to EEG, and whether it's worth how to put it into words, so that also others think that it's worth going to MEG.
So someone else has written in, thank you for a great talk. Do you think looking at other variables such as sleep, quality and quantity, diet, alcohol consumption, smoking, etc. would be useful or could inform the analysis in some way?
Yes, again, very good question. Actually, sleep is very interesting. The thing is that there are some suggestions around the world on kind of sleep differences, or kind of EEG differences, especially during sleep, which could be informative. And we actually have measured a small cohort so that they were really sleeping. We haven't really looked at that data yet, but we actually have now marked our EEG datasets so that sleepiness, because many of our subjects, unfortunately, during these very long measurements, they get sleepy. So we have marked kind of those parts from the data so that we could use them as a separate feature. So at the moment, I don't know whether it's an artifact or whether it's actually an important feature, but we are really going to look at that. I think it's very interesting. In this data cohort, we actually have been trying to make the lifestyle, the subjects from their lifestyle perspective as similar as possible. So for example, if there's a lot of alcohol consumption, a lot of smoking, they are not maybe included in this cohort, but it's otherwise we have certain lifestyle factors that we can then frequent with the data. And I know that, for example, in Madrid, Fernando Maestro's groups, they have been really active in looking at the diet factors, and they are quite nice findings suggesting that it really does make a big difference.
So we have another question. Do resting state biomarkers, say alpha power, change in different directions depending on AD progression stage? For instance, initially increase, then decrease. How will that affect the early detection of AD?
This is also a very important question. There are, before, for example, we put this study together, there were some controversial findings about, for example, this hyperactivity and then hyperactivity in MCI subjects. And most likely, or at least I think so, that could be that these subjects have actually been measured at different kind of phases of the disease. So most likely there is kind of the hyperexcitability start when, for example, connectivity may increase, and then it decreases together with the dying neurons. What we usually see, we see that, for example, the alpha power slows down as the disease progresses, and so on. So there are clear changes, and they are especially clear in the late phases, but especially in the early phases, they most likely are first the hyperexcitability, which may have produced the earlier somewhat different findings. But this is exactly important also, that how can we then how to use this in the early detection. So in that sense, I think it's really now nice that in this study we have the possibility to look at these subjects from multiple measurements, and we also have a lot of this cognitive data which we can then reflect against the MEG-EEG data to kind of get a better view of that, where are we going on in this in the progressing disease.
So even healthy individuals show quite a lot of variability in their electrophysiological signals. What is your opinion on how this variability could be best taken into account in your studies?
I think we are still kind of lacking normative frequency, normative ranges of EEG and MEG data. So if you think about any, let's say, clinical blood value or something, so there's a range which is considered okay and what is considered pathological based on thousands of measurements in different subjects, and we don't still have these kind of things for most of the MEG-EEG features. And I think that's needed. So we would need normative cohorts over different age ranges. There are good cohorts already. For example, the CAMCAN dataset in UK, which was collected years ago and has been very, very useful in this respect. But I think we would need even bigger ones. So we would need to have kind of good ranges of what is normal, what is not normal, and then we could start to look at our patients against these ranges. And also, I am sometimes I think gave an interview here in Helsinki to our main newspaper, saying that everybody who turns 40 should have their baseline EEG or MEG, because of course we are individuals. So if we would have a kind of baseline existing for ourselves, that would be also quite interesting and helpful then for later years when our cognition is maybe not the same anymore, and we could use that as our own references. And I don't think that would be that expensive in the end, taken all the other expenses that we cause.
So another question is, rather than long-term effects of sleep, alcohol, etc., is it important to control for short-term state effects of, e.g., tiredness, hydration, caffeine on the MEEG data? Do you require standard conditions before acquiring data, ex no coffee several hours before, etc.?
We have told the subjects that they are not allowed to drink their morning coffees before they come to the lab. This is very difficult, especially for Finns. But so I don't know whether it's a bigger confounder that they are not allowed to have their coffees. But we try to make sure that they have slept well the previous night, and then they are allowed to have good coffee in our laboratory then after the measurements. But it's really true that there are, we know that these do affect, and we have been trying to like balance these kind of effects.
And our final question, you described here MEG results from both oscillatory activity and task-related responses in MCI subjects. How about aperiodic MEG signals, which have recently gained increasing interest in the neuroimaging community? Have they been studied in MCI?
Yes, well, these aperiodic signals, for example, 1/f signals, have been lately, I think, gaining interest, for example, in Parkinson's disease and in several neuropsychiatric diseases, and they also actually appear really heritable, which is also very interesting from the brain point of view. And actually, we have started to look at those in our MEG data set, and our preliminary results suggest that this could be actually really informative and good additive feature to be added to our feature package to be explored. But we are kind of in the early stages with that, but definitely worth looking at.
Well, thank you so much, Hanna, for a great masterclass today, and thank you everyone for joining online and sending in your questions as well. Thank you.
Thank you.